Good morning, students. Yesterday in our previous class we have discussed about the process of photosynthesis as a mode of autotrophic nutrition which is done by green plants. Now in our today class we are going to discuss about some of the experiments which is here virtually shown by the diagram in our virtual lab. I am just showing you the apparatus, their name, and how are we doing the experiments. When we have our offline class, that time we will do it in our practical. So students, here by this activities we are going to show that the process of photosynthesis. First activity, let's see the first activity which demonstrates that starch is formed during the process of photosynthesis. Students, yesterday we have discussed that by the process the food is formed as glucose, and the glucose is stored in the plant body in the form of starch. So starch formed in the plant body is a result of photosynthesis, and in this activity we are going to do some test with iodine solution which turns blue color of iodine into black by the presence of starch. So students, for this activity, the material required are the 2 potted plants, a beaker, test tube, water, burner, dropper, ethyl alcohol, iodine solution, and glass plate. You can see here what we are going to do in the activity. At first, there is a need to take 2 green potted plants and we will label them as A and B. We will put them in a dark place for 2 days. After that, we will keeping them for 2 days in a dark place makes the leaves starch free. Why it happens starch free? Because no sunlight is there in the dark place. In the absence of sunlight, the leaves are not preparing their food. They will not do photosynthesis. So students, we are keeping them in dark place. After that, we will take a plant and keep it in the dark place, remain it there, and remove the plant B, take out from there and keep it in sunlight at least for 4 to 5 hours. Then by doing so, we will pluck one leaf from plant A which was kept in a dark place, and then one leaf from the plant B which is kept for 4 to 5 hours in sunlight, and we will mark them as A and B respectively. After that students, we will take a beaker and keep the leaf in it and boil it for 2 to 3 minutes. You can see here the beaker is there. This is the tripod stand. This is burner. This burner, we can take this burner, allow the boiling of water, and the leaf is already kept here. After boiling it for 2-3 minutes, we will take out the leaf and keep it in a test tube filled with alcohol and heat them. While we are heating it in alcohol, this alcohol decolorized the leaf color, and it will show burning alcohol. We will heat the leaf kept in test tube for 4 to 5 minutes with in alcohol which destroy the chlorophyll of leaf. So the color of leaf will not be green. We will take it out from it, wash them in warm water and remove the alcohol from it. Then what will we do? We will place both the leaves A and B on a glass plate and put few drops of iodine on it. Here you can see the leaf A is kept and leaf B is kept. On leaf A we will put the iodine solution and B also. We will observe students that leaf A will be same, no change in color of iodine, while the leaf B will show the changing of blue color of iodine into black. Why it happens? Because the starch is present in leaf B. As leaf B is kept for 4 to 5 hours in sunlight, and in sunlight it has prepared its food. So our observation is leaf B turns blue-black in color, but the leaf A does not. Why it happens? As a result, plant B has starch and it was kept in sunlight, which shows that starch is formed only in the presence of sunlight. The process takes place in sunlight, whereas the plant A does not give the test with iodine solution because it was kept in dark place and not done the process of photosynthesis to make starch. Have you all understood this activity? Now let's see another activity. This activity shows that sunlight is essential for photosynthesis process. So for showing as a necessity of sunlight for this process, what will we do? We will first take a potted plant, piece of black paper, clips, beaker, burner, test tube, dropper, water, alcohol and iodine solution. And for this activity, we will keep a green potted plant in a dark place for 48 hours. Again, why? To de-starch its leaves so that no photosynthesis takes place and no food is formed. After 2 days or after keeping it in 48 hours, we will pluck one leaf and test it for the starch, which confirm that the plant is de-starched. We will cover a part of healthy leaf on both the sides with a strip of black paper. You can see this is a leaf, healthy leaf. We are covering it with a black paper strip. Here are the clips which are used to fix the black paper on the leaf, and the black paper is the portion where black paper is kept. No sunlight enters, so no process of photosynthesis takes place, whereas the region which is exposed to sunlight shows the formation of food due to presence of sunlight and doing the process of photosynthesis. After that, we will place the plant in sunlight for at least 4 hours or more. We will pluck the leaf, remove the black paper strip, then boil the leaf first in water and then in alcohol. Again, why are we boiling it in alcohol? So that the chlorophyll gets destroyed and the leaf become colorless. So after that, we will wash the leaf with warm water and we will put it in a petri dish. We will pour few drops of iodine solution on the leaf. What will we observe by pouring iodine solution on it? We will see that the area which was covered with the black paper do not show any change in iodine color. It remains blue, whereas the portion which was exposed to sunlight gives the confirmatory test and iodine turns blue-black due to presence of starch over there. It was exposed to sunlight, so it has prepared food by the process of photosynthesis. What is our observation? The uncovered part of leaf was exposed to sunlight and turns blue-black in color. By testing with iodine, it gives the change in color, while the part of leaf covered with the black paper strip does not give any test with iodine solution. And by this, we conclude our result as starch is formed only in those parts of leaf where sunlight was exposed, and proof that sunlight is necessary for the process of photosynthesis. In the third activity, we are going to discuss that chlorophyll is necessary for the process of photosynthesis. Students, for this experiment, we can take a plant which shows variegated leaf. Means that you might have seen the leaf, the color of leaf some plant like croton, which is generally used for ornamental purpose. It shows yellow dotted in the green color leaf. So we can take that. We can take the leaf of that croton plant, burner, test tube, dropper, petri dish, water, paper, alcohol, iodine solution, and glass plate. Here we will first take the plant of a croton and showing a variegated leaves. Then we will keep it in sunlight for few hours. Then we will pluck the leaf again and boil it in a we will plug the leaf and boil the leaf in in water. Then we will take it and boil it in alcohol. Alcohol, again, what happen? It decolorizes the green color of leaf as it destroys the chlorophyll. After that, we will take the leaf and keep it in a petri dish and put few drops of iodine solution in it. What will we observe, students? That the portion where the green color was there in leaf, it shows it only gives the test with iodine and changes the color of blue color of iodine into black one, whereas the portion which shows the yellow color or white patches of the croton leaf do not give any test with iodine. And we observe by this activity that the region marked green in the leaf shows blue black color here with iodine solution, and the non-green region, which is variegated portion, which is yellow patches, which is for giving a good appearance as an ornamental plant, is remains unchanged. Why? Because it has not done the process of photosynthesis, as it is not green in color. So it means starch is formed in green part of leaf only, where the photosynthesis takes place. Here, by the experiment, we conclude our result as chlorophyll is also essential for the process of photosynthesis. Students, this is all for today. Be safe and remain at your home. Revise the topic, and we will meet in the next class, where we will discuss about another mode of nutrition, that is heterotrophic mode of nutrition. So students, please go through all these experiments, write it in your notebook, and we will meet again. So thank you and have a nice day.